my experience in these topics is limited, and I can make mistakes because I don't follow the all the data that is coming, and there is a lot of data on rapamycin and stuff. So, but according to my model, so we have aging that this is a problem that kills us, but it has some plasticity because sometimes the specifically in small animals. So like let's say if small animals live for a couple of weeks, then these couple of weeks are coming to the heat wave in Belgrade. Yeah. They cannot reproduce during these 2 weeks, and they are all done, right? So in this case, aging would be very damaging for them. So if this damage will be much higher than the benefit of disease control. So this that's why if environment isn't good for reproduction, they need to stop it and wait until the heat wave is over. Then they can aging and die in a couple of weeks as programmed. So this is called aging, plasticity of aging. And I think most of the drugs that we have, they are affecting either this plasticity, that is like environmental, or part of plasticity which is associated with the infections and chronic inflammation. And it may be a coincidence, but I believe it's true. Rapamycin is a immunosuppressor drug. So that's I believe that why it works. So these things definitely they can work, but this is limited. We will never solve the entire problem with rapamycin. This is very clear. Like nobody solved this problem with healthy lifestyle and diet. I have my own opinion, which is different from the field. There is a very peculiar state of cells that they undergo, they enter this state in response to DNA damage or telomere shortening. Telomere shortening, telomeres are caps on the ends of the chromosomes. Each time a mammalian cell divides, these caps are getting smaller and smaller. Telomeres getting shorter and shorter. If they are too short, then the cell is also going into the senescence state. And these caps then in germline, when we build a new embryos, these caps are increased by an enzyme called telomerase. Yeah, but if they are in somatic cells that are building most of our bodies and don't go in the embryo, so they have most of them have this limit of divisions. If this limit is reached, then the cell becomes senescent. It doesn't divide anymore, but it also doesn't die. It is metabolically active. It sits there and do some secretions. So it secretes what is called senescence-associated secretory phenotype, or SASP. So, and this SASP contains of pro-inflammatory cytokines that are damaging for the surrounding. Cells were shown to be contributing to many age-related diseases. And removal of these cells seem to be beneficial for health. So the real biological function of these cells, why are they good for, are not clear. So people believe that they are there to like be induced and then killed by the immune system, and that's what typically happens in young organisms. In old organisms, immune system isn't good in killing them, and then they start to accumulate, making troubles. So according to my model, and we can test it experimentally, that's what we are mostly working on senescent cells right now, not the systemic immunity. So we believe that these cells are antiviral. So these are also part of the immune system. And they can be induced by viruses, and they can also, with this SASP, they can inhibit virus replication. So, and this has been proposed in the literature, but people, I think, do not appreciate it to the extent they should. So, and I believe these are also sort of part of the immunity that later got additional side work in killing organisms in the context of adaptive aging. So potentially removal of these cells, according to my model, should be very beneficial. The problem is that several studies showed that very effective removal isn't really beneficial for lifespan. So that even very strong, so because in people we need to go with senolytics, which are drugs that kill senescent cells, but in mice we can do like strong genetic constructs that allows to do it much stronger, but there they didn't get good results on lifespan. So this so far discourages many scientists from senolytic approach to aging. So, but still, it can be that something is was not like exact in these experiments. Maybe there are some other something is missing, and some modalities where senescent cells removal of senescent cells can provide a strong effect. I think maybe ability to deliver things into cells for sure, like with high efficiency. AI definitely changing our life. So I want to share with your audience an excellent tool called undermined.ai that changed my life recently, because it allows to search for scientific papers in a very effective way. So it is like ChatGPT, but trained on scientific the body of scientific research, and it doesn't make mistakes like ChatGPT, and is not trying to fill gaps as ChatGPT often does. So if you ask it, and there is no evidence for it in the literature, ChatGPT is still trying to imitate. And this undermined says no, there is no good evidence for that. Which so it really saves me a lot of time with the working with literature, and maybe these technologies of this type can build like artificial scientists that would in automatic regime propose hypotheses and like trying to fit them into the existing body of literature. I think this can be the fastest progress that we can probably foresee in a couple years. So the other stuff, I'm not that sure. Maybe building tissues, building organs in vitro, specifically for immune tissues, like making young thymuses or young bone marrows. But also, if you think about rejuvenation of the immune system, right? If my model is correct, aging is not a loss of function phenotype. It's not that something is missing from the old organism. It's a gain of function. That something emerges in the old organism. And if you know this 2019 Conboy Lab parabiosis experiment, right? If aging is something we gain, then we not only need to put in young immune system, right? But we also need to eliminate the old immune system. And this is not very clear how to do. So we need to specifically kill or reprogram some cell types in the immune system, or maybe in the organs that these organs that are building the like bone marrow. There is a stroma, stromal cells that are sitting there,